My name is Priscilla Burns. I'm a student at Towson University, and today I'm going to be discussing an experiment I conducted in my Bio 200 class under my lab under Dr. Larry Wimmers, as well as help from my two other fellow students, Alex Goldstein and Scott Niles. In our gut microbiome experiment, we examined the gut microbiomes of two different dogs. We selected two German shepherds. They both had the similar age of 1 year old. They both got regular exercise. They were both healthy, and they both were barely over 100 pounds. And then the only thing that differed between the two dogs was their diets. One was fed a diet with a lot more starches, and then the other was fed a diet that was very protein heavy and was grain free. That was the primary difference that the two dogs had, so we were expecting to see that be the primary cause of any difference in their microbiomes. So microbiomes are they're basically the DNA profile of all of the bacteria that are present in the gut. We were specifically looking at gut or bacteria out of the lower intestine because we were using fecal matter. The gut microbiome can tell you about a lot of different diseases or potential aids in digestion that people could have based on what profile of bacteria they have. It's been known to even influence factors such as autism, psychological disorders, dieting habits, weight loss, weight gain. So it influences a lot of stuff, and we still haven't even done enough research to know everything that the biome affects or how it affects. But it affects a lot of different health concerns. And so we can then look at these animals and look at the gut, the bacteria that are present in their gut, and maybe make some inferential observations about their health based on what DNA from the bacteria that we find in their gut. So, our question was, how will the gut microbiomes differ between these two animals? So we picked two very, very similar animals to begin with because we wanted to make them as similar as we possibly could, because we really, really wanted to hone into what differences could be present in those dogs and how that could be affecting them. So our whole process of extracting this DNA and then sequencing it started with the use of a Keogen kit. So that kit is designed to separate the DNA from all of the other enzymes and gunk that's in the fecal matter that could be impeding or contaminating the DNA that we're trying to extract. So the Keogen kit was used first, then we used a Zymo kit. Zymo kit is designed to concentrate the DNA so that we then can see actually how much we have to work with and then make the decision of whether or not it was enough to even begin sequencing. Luckily, both of our samples were very, very sufficient. I think the goal read was 1.5, and our number 403 had a 1.86, and number 521 had a ratio of 1.53, and that was done with with a UV spectrophotometry kit. We used a ratio of 260 to 280. And that was an ideal that would tell us how much bacterial DNA was present, and then also if there were any contaminants present. Our numbers indicated that we not only had plenty of bacteria or DNA to sequence, but we also didn't have any contamination. So we were good to go and proceeding. We went ahead and moved on to Amplicon PCR, which is a process that amplifies the DNA. So we didn't have enough to start out with this tiny little sample, and basically when we stick it in the Amplicon, it sticks in a whole bunch of different nucleotides and it starts sequencing more and more of that DNA. So we essentially amplify it and do a bigger sample so that we have more to look at. After the Amplicon PCR, we did gel electrophoresis. That was to tell us if the Amplicon actually worked. It was also to tell us if we had any contamination. So we had a negative control and a positive control present that we ran tests on in the gel electrophoresis. Our positive control was DNA extracted from E. coli, and then our negative control, we obviously didn't put any DNA into at all. We just wanted to make sure that we didn't mess up and get any other DNA into our other tubes while we were running the test. So the negative control essentially just told us that we didn't have any cross-contamination. So our gel electrophoresis was successful. We had plenty of bacterial DNA positive bars above both our samples 521 and sample 406. And then we also had plenty of bars above our sample number, or our positive control. And then our negative control had 0 bars, so it didn't actually develop any DNA at all, which is comforting because it tells us that we didn't mess up and there was no cross-contamination. We then used a PCR cleanup kit. That was designed to pull out any unused buffer or any unused primers. So those primers were the nucleotides that we had mixed in with the amplicon reaction in order to basically be the building blocks for more DNA. And the PCR cleanup removes any excess unused ones of those that could potentially mess up our results. And it also removes the buffer that we used. We then indexed, and the indexing PCR reaction was designed to put little flags, essentially, on the end of all of the DNA that we had collected for our dogs, so that we then could put it in a giant, really expensive sequencing machine that we could tell which DNA was ours in and tell which DNA was from other classmates' reactions. So it's literally just indexing like in a library to be able to tell which DNA was ours. So after indexing, we did the PCR cleanup second round. That was just to make sure that we didn't have any other stuff left in there, any other nucleotides or little bits that could have been left over from the indexing reaction as well. Just wanted to clean that DNA as much as possible. And then we did a fluorimetry test on it. The fluorimetry test was designed to be similar to the UV spectrophotometry. It tells us what the concentration of DNA was, but it also tells us what it was. It's way more specific than the UV spectrophotometry. So what the fluorimetry does is it tells us how much of it is bacterial DNA and how much of it is RNA or other proteins and stuff that we don't want in there, stuff that would be a contaminant. Our fluorimetry test came back clean. We were not contaminated at all in either of our samples. So we then went on to sequencing. The sequencing was done by the lab instructors or by whoever, I guess, carried that out. We didn't do it personally ourselves, but that ended up giving us results with a company that I can't remember the name of. But we got a really big data sheet of all the different proportions of different bacterias or really any organism DNA present at all the different levels. So from kingdom all the way down to species, we got pie charts and percentages of what DNA was most dominantly present in the fecal matter at those different stages of, or those different levels of organization of life. So we then moved on to examining these. So we looked at all of our numbers. We had really, really good read numbers for both of our samples. Both 521 and 406 were over 7,000. 521 was actually at 13,000. Our positive control actually was only at 3,000 with the reads, which is above the threshold, but it's still lower than our other samples, so that wasn't ideal. And then our positive control also, it looked really good until we got down to about the order level and the genus level. We started seeing some other contaminants of the Serratia bacteria. Every single other group aside from one in the lab also had this contaminant in their positive control. So we then can assume that this happened before the lab was conducted, possibly even contaminated at the original lab that isolated the DNA before it was distributed to us. So on top of that, we all of our results were really good. We got some really similar DNA present in both 521 and 406 right up until about the genus level.